Well, time out, slow down. Now, it's, the truth is important here, Marilee. Okay.
Yeah,
So, 80% of the
yeah,
microbes in the world live in a biofilm.
yeah, exactly. So
Oh,
on
man.
rock, on tree, on everything, I think.
I slept through that lecture in microbiology. Okay. So, 80,
I
so
mean,
bio,
Matt, think about what happens to your teeth when you wake up
Yeah,
in the morning and you haven't brushed your teeth in 12 hours.
they smell
That,
bad.
yeah, that like
that is
film,
biofilm.
that is a biofilm. This is just how bacteria like to survive. They like to live in biofilms.
At Dairy Digressions, we talk to the people behind the latest discoveries in the dairy industry. If you would like to connect with those people, then the American Dairy Science Association is for you. The ADSA is where researchers, educators, producers, and leaders in the dairy industry come together. Join us and become a member today at adsa.org. Hello and welcome to a special episode of Dairy Digressions, a podcast of the American Dairy Science Association, the Journal of Dairy Science, and JDS Communications. I'm Matt Luce, former editor-in-chief of Journal of Dairy Science and former editor-in-chief of JDS Communications and fellow of the American Dairy Science Association. I'll be your host today. Dairy Digressions is where we get an inside look at the science and scientists making headlines both inside and outside the dairy industry. We expose the latest research that is creating knowledge and drives innovation in the production and processing of milk for human consumption. Today, I am honored to be joined by a guest co-host and special friend, Nicole Martin, who is an assistant research professor in dairy foods microbiology and associate director of milk quality improvement in the Department of Food Science at Cornell University. She's a senior editor for the Journal of Dairy Science and a member of the ADSA Board of Directors. Welcome, Nicole.
Thank you so much. I'm very happy to be here with you.
It's awesome to have you. I love to hear your very energetic voice. You're telling me, and we're still making ice cream at Cornell, right?
We're ramping up ice cream. The students are
Oh,
back. They eat a lot of ice cream.
you can just buy it from Penn State and just like sell it.
I do buy it from Penn State when I'm in Pennsylvania, but here in New York, it's all Cornell all the way.
Okay, Cornell, the Big Red, the Big
That's
Red
right.
Dairy Barn, and so and we have two special guests today. We're going to talk about a biofilm special issue that's coming out in the Journal of Dairy Science. Marilee Gagnon and a second special guest, Tara Wilson, and I'm going to tell you about them individually. I'll start with Marilee, and she is super. She's got a bachelor's degrees in biology from the University of Quebec. Oh boy, Chicoutimi, yeah. Okay, everybody, a little geography lesson. This is sort of just a few hours north of the St. Lawrence, right? Sort of
Yeah.
in Ontario. It must be in Ontario.
No. It's in the north. You have a little river. It's called Saguenay,
Oh, yeah. Okay, so
and
it's in Quebec then. It's in Quebec.
it's in Quebec. Yeah.
Oh yeah, fantastic! Of course, you speak French. Yes, she has. She's currently at the Acer Center as a scientist, and she's going to tell us about that. Before that, she was a lecturer at the University of Laval. Is that correct, Marilee?
Yeah, exactly.
But you're going to tell us a little bit more about the Acer Center when we get into the discussion today. And
Yeah, a little bit.
our second guest speaker is a PhD in food science, and she's at the University of Guelph. Tara Wilson, welcome, Tara.
Thanks for having me.
We love the University of Guelph at the Journal of Dairy Science and ADSA. It's one of our major contributors to the journal, to both journals. Shout out to Steve LeBlanc and everybody at the University of Guelph. Tara is a PhD student with Gazelle Le Pont, and she's in food science. I think I'll give you a little interesting thing about Tara. She has an undergraduate degree from Lakehead University. And if you don't know where Lakehead University, that is like north of Thunder Bay, correct?
Yeah, it's right in Thunder Bay, but we're way up there, that's for sure.
Yeah, that is like God's country up there. So that's sort of just north of it's north and west of Lake Superior. I don't know if it's on Lake Superior, Thunder Bay.
Yep, it's right on the lake. I used to be able to walk out of my apartment and see the sleeping giant in the background, so that was pretty nice.
Yeah, so that is just a beautiful part of Canada, and that's still in Ontario, though, correct?
Yes.
Yeah, so, and then from Lakehead, it looks like you moved to the University of Guelph.
Yep.
how'd you get interested? And just as a student, I'll just kind of shoot this in here. How'd you get interested in food science?
I actually took biology of food safety course when I was up at Lakehead with a former professor for the University of Guelph, Heidi Schraft, and she directed me to Guelph and said, I think you'll have the best time of your life there. And I never looked back after that.
That is fantastic. Yeah, Guelph is a great institution. There's no like dairy cows in Thunder Bay, are there? They would freeze up there.
There is no dairy cows in Thunder Bay.
Okay. I'm just saying, the wind comes off that lake. It's got to be a little. No, it doesn't really come off the lake. The wind must go onto the lake. Okay,
The wind
but I'm
goes
not
onto
sure.
the lake. Yeah, but
Okay, yeah, but it's a bit
very cold.
So why are we here today? Well, we're here to talk about a special issue on biofilms in dairy, in dairy, both on dairy farms and dairy processing plants. And this was a topic that was just super new to me. And I didn't know that much about biofilms and how that all works. I do know a little bit about microbiology, but I didn't understand that these little guys, these little microbes, which are all around us, also create a lot of challenges, particularly on farms and on the food processing side. And these three are experts on some of these challenges. And so Denis Roy and Marilee Gagnon made a proposal to write sort of a special issue. And what a special issue is a series of articles that deal with some of these challenges of biofilms. And I think that's really great. And I want to remind everybody that the Journal of Dairy Science is 100% open access. And what that means is you can access these articles free of charge. There's no paywall. You're welcome to look at them, read them, and explore this topic further. So there's a series of articles, and Marilee co-authored sort of an introduction, and then Tara co-authored sort of a paper on kind of tracking it, these biofilms all the way from the farm to the factory, the processing plant. Where are they? There's another paper. Oh boy, Tara's or Marilee's going to have to help me on Chloe Desmusu. Anywhere close? No.
Demuso.
Okay, yeah. And that's on sort of laboratory scale studies of biofilms, which was super interesting. Marilee, you have one on Pseudomonas and yeast. I guess these are two important microorganisms that we think about. And then Coralie Getz has an article on sort of new processes to try to combat these things in facilities. And that's important because a lot of the processes we use to kind of combat biofilms are kind of old school, like acid washes and stuff like that, and maybe Nicole eventually got to step into the modern era with something other than an acid wash, right?
An acid wash applied correctly is a very powerful thing. So.
Okay. So maybe we'll talk about that as well. Okay. And then finally, there's a really interesting article by Elodie Desmontier. Close?
Desmontier.
Yeah, close. Yeah. And she's looking at, a lot of times we think about microorganisms like Staph aureus as just Staph aureus, but actually what's super interesting about microorganisms is it might be Staph aureus, but it may carry one gene or another. And she's talking about these staphylococcal protein A virulence factors and their implications. And so that's a really deep dive into some microbiology. So there's a great series of articles. And so we're going to take a few minutes to discuss these articles with our guest speakers today. And I'll handle some questions and Nicole will do some questions and we'll sort of do that. So the first question that I have is, I don't really know anything about biofilms, okay? And they are nasty little things. So Marilee, can you just tell us a little bit about biofilms and specifically talk about, kind of where they come from and, what is a biofilm in dairy when you think about it?
It will be my pleasure. So biofilms are basically a lifestyle for microorganisms that allow them to attach to a surface and grow in community. So interestingly, more than 80% of the microbial biomass on our planet actually exists in form of biofilms. So you didn't know that for sure.
Well, time out, slow down. Now, it's, the truth is important here, Marilee. Okay. Yeah. So 80% of the microbes in the world live in a biofilm?
Yeah, exactly. So on rock, on tree, on everything, I think.
I slept through that lecture in microbiology. Okay, so
Matt, think about what happens to your teeth when you wake up
Yeah,
in the morning and you haven't brushed your teeth in 12 hours.
They smell
That,
bad.
yeah, that like
that is
film,
biofilm.
that is a biofilm. This is just how bacteria like to survive. They like to live in biofilms.
So I noticed in the articles it was really interesting. You see, you guys, you microbiology people use words I don't understand. Okay, so you talked about planktonic, like planktonic versus, and planktonic is sort of a free floating
Yeah,
organism, and that would not be the norm. It's what you're telling me is planktonic is
exactly.
not right. It's just not. They're just kind of floating. They like to live in these little communities.
Yeah, and the reason is why is because the bacteria will produce a cell matrix that is extracellular polymeric substance, and it will protect them. It's like a layer that will protect them from harsh condition of the environment. So like desiccation, cold, cleaning products, so acid.
Mm.
So this will help. It's why it's their preferential lifestyle. And in the food industry, biofilm will be found almost everywhere, like we said previously. But the most affected sector, I think it's meat, fruit and vegetable, and dairy. And I want to share that during my postdoctoral fellow at Laval University, I work on biofilm and dairy. And now why I'm working in ASAR Center, it's because biofilm are also present in maple syrup industry. So they come get me with my expertise on biofilm. So it's why now I'm working on that.
Which is a bigger industry in Canada, milk or maple syrup? Which one sells more?
I think I don't have number to prove what I'm saying, but
Yeah,
I think it's milk because it's all year long, and maple
yeah,
syrup, it's a small time of the year. So I think it's
yeah.
for
You know, it'd
that.
be really great if you guys made more maple syrup than milk, because then I could harass Stephen LeBlanc about that forever. Okay, like, oh, you're second only to the maple syrup in Canada, but maybe I'll have to investigate that and ask
But
Stephen about that.
interestingly, in Canada, but in Quebec, because Quebec is the more province that produce the most of maple syrup, there's a lot of dairy farmer that also do maple syrup. So,
Yeah.
Yeah,
so I'm
that
like,
makes sense.
That's very interesting about Canada for you people that don't study it is you would think that Ontario was the number one province, and it might be, but Quebec has almost as many cows
yeah,
as Ontario. There's a lot of cows up in Quebec.
yeah, it's smaller cattle than in Ontario in by million in
Yeah,
average.
yeah.
Yeah, but well, yeah.
Yeah, and they speak French up there. Yeah, those cows. So you have this. So maple syrup industry also deals with biofilms. Dairy
Yeah,
industry deals with biofilms. There's biofilms on farms. There's biofilms in processing
yeah,
plants. I was interested when you mentioned, I read one of these articles that there's biofilms inside cows. I mean, that they like to live
yeah.
on. What is that? That is like a biofilm. Okay. Like it's a
Yeah,
double
because
bio.
biofilm can be formed, as we said, on abiotic surfaces like wall, cutting equipment, hooks, pipe, drains. But yeah, they can also grow on biotic surface like dairy meats, sliced fruits and vegetable, and like you said, even in cow's udder, like in the study of Desmontier. In cow's udder is more cause it's pathogenic biofilm that will cause mastitis.
Yeah, yeah. And so it's really interesting. So a lot of times mastitis is hard to treat. And you mentioned in one of those articles that sometimes that has to do with the fact that these things are in biofilms and
Yeah.
the antibiotic just never really gets to the microorganism. I had never really thought about microorganisms living in communities like this. And I guess it's kind of like if we all live together and make certain products, we can make each other happy,
Yeah,
and
exactly.
Yeah,
yeah, and so that's kind of like that. People live in communities too, right? And we sort of have
that's great.
shared things that make everybody happy, and that's really interesting. So when we talk about biofilms, like Are there certain bacteria that are really good at making biofilms, or just the biofilm kingpins of the microbial world, or are they all the same? Or
First of all, I would like to emphasize that biofilm are not only produced by bacteria.
what?
Yeah.
I don't think so now. What do you mean?
Most of research focus on bacterial biofilm, but fungi are also important biofilm producer.
Oh,
For
no.
example, in my study published in the special issue, I showed that yeast, especially Candida parapsilosis, are a major part of biofilm in milking system. So yeah, we need to think about fungi when we plan study on biofilm now.
But yeah,
In
because you wrote a whole
Absolutely.
article on, and you brought up the yeast thing, as well
Yeah,
as was it Pseudomonas and
yeah,
yeast?
and it was really a surprise to me to see so many yeast in the biofilm that we assembled. So
Well,
I
is
was
yeast like, Nicole? Are you got yeast in your ice cream there, or what do you get? You get bread if you get yeast in the ice cream, or what happens?
Well, you have to remember that the product that these organisms get into either support the growth of the organism or don't. So in ice cream in particular, we're not going to get a yeasty sort of off flavor odor because it's not going to grow at that temperature. But in certain circumstances, I've worked with dairy processors who have yeast contamination in milk, and you absolutely open the milk carton and get, but I think more, more so in like lots of styles of cheese, yeast can be spoilage organisms there. So sure, we can get lots of different problems that occur when you have yeast. But I think what Marilee brings up, and I think is really cool, is the sort of research aspect of these, fungal bacterial communities that live together. And often research is done with a single, organism and how it produces a biofilm. And that's just
yeah.
not how things work in nature. And I think the more we can accommodate a more realistic scenario in research in the way that Marilee did with yeast and Pseudomonas, that really helps us to understand these communities a lot better.
It's
Yeah.
important, right? So scientists are reductionist in general, so we try to
Mhm.
study things in isolation, and certainly when we talk about microbiome, that's a community of sorts, and and you separate these organisms apart from one another, and they don't function in the same
Yeah, but
manner.
yeah, exactly. But there are genus species that are especially well known to form biofilm. It is shown in the Musso and Lapointe reviews. Pseudomonas, we talk already a lot about that. Staphylococcus aureus, Streptococcus, Enterococcus faecalis, and Listeria monocytogenes. I'm sure that Tara will talk a little bit more on Listeria. But
Hmm.
these microorganisms can also include non-producer in their biofilm. So this is
Hmm.
thing to keep in mind. And I would like to share a fun fact about my current research on maple syrup. I also found that Pseudomonas are forming biofilm inside the tubing of sap collection system. But
Uh-huh.
in that unique case, Pseudomonas are actually linked to good flavor of maple syrup. So it was really, wow, we love Pseudomonas now.
Yeah, we want good. I thought it came from the tree. You're telling me that bacteria add some good flavor to
Yeah,
the maple syrup?
yeah, exactly.
Oh my
So,
gosh.
That is a new one.
That's a
Yeah.
new one, right? That's a
so
new
you
one.
know, Pseudomonas always smell a little bit fruity, and
Yeah,
Absolutely.
yeah.
yeah, so it's
Really?
that,
It adds
yeah.
something. Oh my
So
gosh.
So it's
Well,
funny because I was sort of raised in old school dairy microbiology, right? And the trick to tell whether you had Pseudomonas growing on an agar plate is to take the top off and smell it, right? And if
Yeah,
it's got
oh
this
yeah,
wow.
fruity smell, then it's likely Pseudomonas. And so, yeah, I mean, in by itself, it's not actually an unpleasant odor. So it's
Oh.
really cool to hear that in certain products that's actually advantageous. That's pretty cool.
Just before I want to keep talking about this because it's so interesting. I never thought I'd be so interested in biofilms, okay? But your hook is this 80% stuff. But so Swiss cheese, is that the result of the holes in Swiss cheese? Is that from yeast? What gives Swiss cheese the holes?
No, that's bacteria.
Oh, darn. I was
Yeah.
hoping that was yeast. Because in bread, it's the yeast that kind of gives it the holes, right?
Yeah.
But not in milk, not in Swiss cheese.
No.
Darn.
Yeast can cause a blowing defect in cheese, and we
Oh,
we run
no.
into that, but it's never, well, I'd say most of the time it's not characterized as a good thing. And my colleague here at Cornell, Sam Alkane, he actually makes cheese with yeast. So it's a, sort of a strategy to avoid problems that we run into with lactic acid bacteria, and there are lactic
Yeah.
acid yeast. So you can actually make a cheese out of yeast, but often it's a defect, not a desirable
Yeah.
thing.
So, Marilee, where we were kind of talking about, and I just want to make sure I don't get out of bounds here, but so Nicole was telling me that there's like 5 miles of stainless steel tubing in a, typical dairy processing plant or something. And so, anyways, the milk is flowing through these tubes and it's all stainless steel and it's all pretty, right? And then, I mean, does the biofilm start with just one bacteria just stick into the stainless steel? Are they all
Yeah.
like brothers and sisters? Well, they don't really mate, but anyways, are they all like sisters of are bacteria male or female? I'm not sure, but are they all
In
bacterial
French,
brothers? Is it all a
it's
single?
female. In French,
It's
it's female.
female. Okay, the bad guys are female. Oh no. So but do they all just sort of does one stick and then starts forming a biofilm and attract others to it? Is that how that goes down?
Yeah, maybe others can correct me, but I think so, yeah. And we saw that a little scratch in stainless steel can be a good point to begin a biofilm because the cleaning will not clean the bacteria in the cracks, so and will start.
Oh yeah, it can be a tiny scratch,
Yeah.
like microscopic scratch
Yeah, and some bacteria are able to form biofilm on glass. So
on glass.
yeah.
Yeah. I think that's one thing that a lot of mammalian biologists don't understand is that, we look at cells and we have our microscopes and we look at them and, relative to a mammalian cell, bacteria are really small
Yeah.
and really numerous.
Exactly.
So you can't sort of just they're small, they're numerous, and they stick in places. Okay, Nicole, you're on.
Yeah, I just want to build on the idea of sort of these different organisms and how biofilms are a community. And Tara, if you could talk about these different organisms and the impact that they have on dairy products, either at the farm or at the processing level, and even at the consumer level, importantly, because if we have these communities and we're getting a variety of microbial contaminants, whether it's fungi or whether it's bacteria, what do they do in that product? How do they impact the finished product, ultimately the consumer?
Yeah, for sure. So it really just depends on the type of bacteria that are in the biofilm. So depending on what it is, if it's a pathogenic bacteria, it can cause concern for human health, while spoilage bacteria, more like Pseudomonas, impacts end products and off flavors and odors and discoloration. But that being said, and this is a little bit of a switch of the narrative here, not all biofilms are necessarily causes for concern, especially when you're looking at processing systems. So for us, we looked at an on-farm producer, which a big priority for them was having biofilms on their cheese ripening boards. And that was a lot of actinobacteria and brevibacteria where they were making a wash rind cheese. So it helped out with their cheese ripening. So
Yeah.
it really just depends on the bacteria and what scenario it's in for how it can impact dairy foods and human health and ultimately the consumer.
Yeah, I think that's a really good point, Tara, because we didn't always have culture companies who supplied our lactic acid bacteria or our fungal organisms, right? And processors, these sort of what we would consider now artisan processors, used the biofilms in their vats and on their aging boards to inoculate the milk with the organisms that made the cheese or the yogurt, right? And controlling those biofilms was really the art form of making cheese or making the cultured product. So I think it's a really good thing for us to keep in mind. But talk a little bit more about, sort of the concern on the pathogen side of things in biofilms and especially finished product after pasteurization.
For sure. So I know one big one that a lot of people like to talk about it is listeria. And depending on the species type, some of them are actually weak biofilm formers and need other bacteria like a rhodococcus to help improve their formation. Whereas sometimes, depending on the temperature and the humidity that it's in, it's able to form biofilms just fine. But a lot of things that we see are sort of the multi-species and cooperative behaviors in between bacteria that, like Mary Lee said, 80% of bacteria are able to form biofilms, but it's whether or not they can form biofilms efficiently that really impacts whether or not it stays in production and processing. And that's where a lot of the multi-species stuff comes in, only because other bacteria can sometimes help weaker formers in order to make a more cohesive biofilm.
And so when we think about the product and the sort of product contact with a biofilm, and I'm mostly referencing sort of post-processing, but obviously this happens pre-processing as well, and it happens on the dairy farm. We talked a little bit about sort of the intrinsic and extrinsic factors of a dairy product and whether that would allow for the growth. And I think my example was ice cream, right? Like we might get contamination in ice cream with yeast, but at the temperatures that ice cream is held at, it's not really going to cause any, quality issues. But talk about the susceptibility of different dairy products to microbial growth and what that looks like. And from the processing side of things, people are really concerned about biofilms. They spend a lot of time thinking about biofilms and where they are and how to find them and that kind of thing. Can you kind of separate the idea of if these organisms that are living in biofilms can grow in the finished product, what kind of products are susceptible, and the presence of biofilms in general, regardless of whether the organism can grow in the finished product?
So again, coming back to just kind of what type of bacteria is in there. If we have things like psychotrophs that really love sort of a cold growing environment, so areas like a bulk tank would be potentially an issue for that type of bacteria. And then even in the processing side, I'm more experienced sort of with cheese processing. I've never really dabbled too much in yogurt or ice cream, so I'm not 100% sure of the biofilm issues that they face, but kind of going along with Mary Lee, for cheese ripening, a lot of times it's done in high humidity and warm temperatures, which oftentimes give a great opportunity for certain bacteria to grow efficiently. So whenever there's sort of an instance of a biofilm, there's always a potential that if the growth conditions become proper, that it can really influence the amount of bacterial growth that there is.
Yeah. Yeah, I guess it comes back to this idea of, do we care? Right? Like if there's a biofilm, but it doesn't contain a bacteria that's going to grow in my finished product, do I care that there's a biofilm?
I think, yeah, absolutely we do. We do still care because it's not to say that the bacteria that's in there currently doesn't cause a problem towards milk. It can also just influence the growth of other bacteria, especially with these multi-species. And like I said, not all bacteria are bad, but there is always a chance that it will contain a bacteria that is bad to either human health or spoilage.
Yeah. Yeah. And I think what we talk a lot about with processors is, sort of using the presence of those biofilms almost as verification of cleaning and sanitation efficiency, right? And so if we have a biofilm, regardless of whether it's, causing a finished product issue or not, or a public health issue or not, it means that something somewhere in our system, we weren't efficiently cleaning and sanitizing our rubber goods or our stainless steel, or maybe we have this niche. I think Marilee talked about, defects in stainless steel and how that allows for the sort of establishment of biofilm. So I think there's a lot of different ways for people to look at it and consider it. But for me, I kind of always come back to, well, if we're getting biofilm formation, then there's probably, at least on, I'm not talking about like cheese boards, but on, fluid milk processing, for example, it means that we need to reconsider how we're doing cleaning and sanitation.
For sure. And the thing with biofilms, they can really attach, they could really go anywhere that they're able to efficiently attach. So like Marilee was saying, this can be seen as a scratch in stainless steel. that gives it a rigid enough environment for attachment. We see it a lot in like cracked and kind of striated hoses and gaskets. So before you even see that there's a major crack in your gasket or your hose, if you're kind of able to bend it and you can see little striations kind of come up, that's an excellent place for kind of bacteria to hide. And it makes the removal by shear forces and sanitizers and washes just that much harder, right?
Yeah, absolutely.
Also, what we need to to keep in mind is that biofilm also can cause biofouling, and then it will be another problem. So in dairy farm, when you have the milking pipe, if there's a biofilm that was not cleaning efficiently, it can grow. I don't know in English, but in French they say they have cheese in the milking pipe, and they will stop the good flow of the cleaning. So we need to keep that also this in mind.
We've been talking a little bit about cleaning, and I want to talk a little bit about how cleaning is done now, and then how cleaning might be done in the future on both dairies and processing plants. But before I do that, I wanted to sort of digress a little bit. Are you ready, you guys?
It's the name of the podcast.
Yeah, dairy digression. I always thought about, when we talk about bacteria, now I know that most of them live in communities. And I always thought about, why does one dairy farm differ from another? Why does one dairy farm different from another dairy farm? And then why are startup dairies, when you have basically a brand new build, the dairy has never been there before, why is that often so difficult? Why is that so difficult and full of surprises? And I've always wondered if, when you have a startup dairy where it's never been there before, new barns, new concrete, new steel, it just sort of takes time to build up a microbial community. And sometimes it can be a very supportive microbial community. And sometimes it just isn't really the right microbial community for those cows, because I think a lot of times we forget that as much as, we're very large organisms as people, and we work with even larger organisms as cows. As they say, the meek will inherit the earth. And these little bacteria have a tremendous influence on everything around them. And even though they're small, they're quite mighty in number, and it also brings me back to a visit. I was in the Parma region of Italy, and I was touring a plant that was making Parmigiano Reggiano, and the importance of those bacterial cultures, which have been around for a long time, and the importance of the farms where that use that milk and the production of that cheese, and the importance of what those cows ate with respect to the production of that cheese. And I can't remember, but there was some sort of rule that can't eat corn silage because that'll mess up the cheese, and like, what do you mean that can't eat corn silage in America? 50% corn silage, when you start to sort of really appreciate a lot of what you guys have been talking about today, that a lot of cheese production depends on the good guys and the bad guys. It depends on the good guys, and I think on our list we had this one for Tara, and so we're talking a little bit about sanitizing, and of course on dairies, as most of you, hopefully people understand, I believe all that dairy equipment with all the milking and the piping, I know that sanitized at least once daily, and probably some dairies milk 24 hours a day, but they always have to have a downtime to really wash that. And I think that's usually an acid wash of some kind. I guess this is for Tara, is like acid the go-to thing for you guys? Like what for washing, or is it acid and I don't know, Dawn soap, or what the heck you put in those pipes? Anyways, I like Dawn. It's good for hand soap, right? Acid.
So, like Nicole said before, an acid wash used properly can be extremely efficient. And it also kind of depends on what stage the biofilm is in. There's multiple different stages, usually around 5. And when it's in sort of the reversible attachment phase, things like shear forces designed by your slugs and everything else like that can really remove the bacteria from a surface like a stainless steel pipe. But as they attach and hunker down a little bit more, especially with their matrix, you really need to introduce things like acids and sanitizers to efficiently remove them.
Yeah. Just what is a slug? You said your slug. I mean, is that like, there's not like those little slugs like you see in the garden inside the pipe. Is there? Is that, what's a slug?
No, it's just when your water really goes through your pipes and it creates a lot of shear force around the whole inside of
Uh,
the pipe to really get the whole pipe clean rather than if you think about just water moving through a river, it usually stays on the bottom sides, whereas a slug is meant to coat the entire pipe.
right. So get some force in there to really force them off. And so
Yeah.
it's always the nooks and crannies that get you, right? Though even with acid washing or slugs, it's either a scratch or even a pipe connection that gets you. Is that what gets, what's your nightmare? What's your nightmare in your, on your dairy or on your, in your processing plant?
Yeah, so definitely little nooks and crannies like that. Like I said, if there's any sort of cracks in hoses, that's a big problem for us. And then another thing that we kind of see is the slope of the milking system can actually make a
Mm.
huge impact. So if it doesn't have a good slope and there's residual water sitting in the milking system after cleaning, that's a huge thing for us.
A
And that
good
can cause a
slope.
lot of problem.
Well, they like I walk in watching the dairy processing plants, all those things are like horizontal. Is that so you want a little bit of slope on there or what?
Believe it or not, they look horizontal, but there is slope that kind of works towards moving them so that there's not just residual water sitting there all the time.
I'll have to bring in my little I don't know, my level, my carpenter's level and check your pipes, okay? Is that so? Let me guess, Nicole, you got a little carpenter's level or something? You check people's slope on your pipes or what?
I do. I carry a lot of things in my tool belt when I go to farms and to processing facilities, and definitely a level is part of that.
No, you're kidding, right?
I'm
Now this
totally kidding. No, but a flashlight, you cannot replace a good flashlight, regardless of which facility you're going into. You have to have a good flashlight. Keep that in your pocket. I think Tara brings up some really good points on biofilms. For me, it's always like, let's do the simple things really, really well. Like, let's do preventative maintenance. Let's make sure that our CIP chemicals are in the right concentration. Let's make sure our water temperature is hot enough, right? Like, those things don't cost a lot of money. They're not like cool and sexy and new innovative things. They're just like the really basics of cleaning and
Like
sanitizing.
testing
Yeah.
water once a year. If you have a lot of E. coli in your water, they can maybe produce biofilm when you're cleaning with that water. So it's a
clean
simple thing
water.
Yeah,
you need.
absolutely. We could do a whole podcast on water and what's in water, but you're so right. Microbes in water exist as biofilms. That's absolutely what happens. Pseudomonas is super prevalent in water, right? And,
Yeah.
and lots of other things too. It's not just, sort of coliforms and E. coli and the things that we traditionally test for. The water can absolutely be a source of microorganisms because of this
Yeah.
you know, massive biofilm formation in these
Right.
systems for sure.
And speaking of nightmare, for me, the brush in automatic milking system, I think it's really a nightmare for me because they clean the cow udder and stuck will the fecal matter will be stick on the brush, and if the brush is not cleaning well, so it will be from a cow to another cow to another cow. So for
That's
me,
interesting, right? Those brushes are that's
you
correct,
you need to make sure that your brush are clean.
Merrily, you bring that up. I say, I at home, I used to have this brush I love to clean out my coffee thermos with, and my wife would always throw away my brush, and I'm like, "Why are you throwing away my brush?" And then she said, "It's disgusting." Okay. I didn't know my wife was a shout out to Jackie, microbiologist. So, Marilee,
Who
you're not into
knows?
these brushes at all.
Yeah.
No way, huh? Oh, that is so cool. And I was, as you guys were talking, I love talking to food people because it's so, I don't know, so different from the production side of things. But so I'll ask this to Nicole, when you see a startup processing plant and obviously all new stainless steel, everything's new, do you see a progression of problems developing over time that might indicate that over time that brand new stainless steel is sort of developing some biofilms, or is it not like that?
So yeah, I mean, I've seen this in numerous cases, but I think what it's about is how well can you control that system? It can be a small system and really well controllable, meaning that you can make sure you don't have any defects in your stainless steel, that you're doing your preventative maintenance. One of the big challenges with these massive processing facilities, when they're new, that's great, but keeping up with preventative maintenance, changing out all the gaskets, making sure that, there are no defects in the stainless steel, it's almost impossible, right? Like it's really, really a big challenge. So it doesn't matter what size the facility is, though, if you're not intentionally making sure that everything is going correctly, then eventually you're going to get problems, right? And you may have really great success for a year, 6 months. It doesn't, like the
Mm.
time varies, but eventually things are going to materials start to get defects. Things are going to start breaking down. If you're not using the right cleaning chemicals, it can actually cause pitting in your stainless steel if you're not using the right temperatures. So paying attention to all those little details, and that's why I always say, like, take it back to the basics, right? Make sure all your temperatures are right. Make sure that your concentrations are right. Make sure that you're working with your chemical company to dial in the parameters that are right for your system, because bacteria are going to find a foothold. And milk comes into the processing facility with bacteria in it. It's not a sterile medium, right? They're gonna be there. They're in the environment. We talked about listeria earlier. Listeria is an environmental pathogen. It lives in drains, it lives on floors, it lives, in the little puddles of water that are left in processing facilities. So they're all over the place. And our job is to prevent bacteria and the yeast and the fungi from getting a foothold in that equipment. So if you're focusing on all those things, all the basics, then you have a much higher likelihood of preventing that contamination in the first place.
Yeah. One of the things a term that Nicole used that some of our cow guys might not know about, CIP, which is clean in place.
There you
And
go.
that just basically means that for a lot of our piping and vats and everything else, you clean in place means you don't tear them all apart and spray them down with a hose or something like that, that you actually are flushing some sort of solution through it, and it's not all disassembled normally.
Right. And I think what's really important is CIP is easy in the sense that you don't have to tear everything down and manually scrub it, right?
Yeah.
But a lot of times people put a lot of faith in their CIP systems to just control things, right? And we can't underestimate just visually inspecting our equipment, looking at it for defects or for biofilm formation. You can't always see biofilm formation, but sometimes you can. Sometimes it gets bad enough that you can. And a lot of times what I see being problem is that we have CIP-able equipment, right? Clean in place equipment, and we have COP, clean out of place equipment.
Oh, okay.
And a lot of times people will try to keep their COP equipment still in line when they're doing their CIP as like a, labor savings or a time savings kind of thing, right? Like, I'm not going to take that out this time. I'm going to leave it in. But that's how problems start, right? When you're not doing those basics really, really well.
Right. So as we kind of draw to a close here, we want to ask about the future. And Nicole, you were going to kind of discuss this with our speakers or ask a few questions about the futures.
Yeah.
What's next in controlling biofilms?
Yeah, absolutely. What's next in controlling biofilms? What tools exist out there to help processors find biofilms? Or Matt was referencing the miles worth of, stainless steel that are in processing facilities. How do we find these things? What are the tools that processors can use, or producers, right? Because biofilms happen in, Tara was talking about biofilms at the farm level. How can we narrow down where those things are? What kind of tools exist for folks to use to identify where they are so they can get rid of them? Marilee, if you want to go ahead, and then we'll give Tara a chance to jump in too, if she has any thoughts on that.
For sure. I think, as you said, detecting biofilm quickly is really the key, but it's not always that simple. So the tool that everybody knows is the swabbing, but sometimes it's not often enough to detach the biofilm from the surfaces.
And when you say swabbing, are you talking ATP swabbing, or are you talking
No,
like swabbing and then culturing?
swabbing and culturing. But I think it will be the same problem with ATP swabbing, that if you not detach
Getting them,
the cells, you will not detect the fluorescence. So I think this stayed the same problem. But a study of Nibusha and colleague in 2020 published an interesting study that combined a synthetic sponge with sonication. So what they do is they use a sonic toothbrush and put a synthetic sponge on it, and this will help to remove the cells. So you can use this in the industry to remove. But like you said, Nicole, you need to do analysis like culture or qPCR. So this will be time consuming. So ATP metry can help. But also in that point in Tara reviews, they talk about UV detection. Maybe Tara, you can talk about that.
Yeah, for sure. So we've actually seen a couple companies now. They're really prominent in the medical and pharmaceutical industries, and they actually have lights that are being designed to detect biofilm formation, which would be beneficial in a system like a dairy system where, like you said, we have 5 miles of stainless steel pipe, and nobody wants to swab 5 miles of pipe and hope that you get what you're looking for, what you're trying to avoid. And even with these things being developed, they're very good at picking up residues. And whenever you have milk residues and protein residues on a surface, it's kind of an indicator that your wash isn't washing properly. So even if they're not 100% designed for biofilms, a light like that indicates kind of how efficient your cleaning is going.
Yeah,
for sure.
Yeah, and I think removing milk residue quickly as possible will prevent the presence of a conditioning layer, because some bacteria could be more easy for them to grow biofilm on milk protein than on the stainless steel surface, for example.
Yeah.
There are also a promising approach to perform continuously monitoring of biofilm by tracking parameters that biofilm influence, such as heat transfer efficiency or electrochemical signals. So in the facility, if you saw that the heat transfer is lower, you can say, oh, my cleaning is not good, I will do a cleaning, and this will remove the biofilm. I also think that integrate AI in analyzing the data and predict biofilm formation will be the future of the large facility. So you have more risk to have biofilm in this room with this equipment. So, okay, we will add more extra cleaning for this one.
Yeah, I think especially on these large facilities where, it's so difficult just to keep up with preventative maintenance and those kind of things, having a risk-based approach, using some machine learning tools that can help target toward where are we going to spend more of our energy, more of our resources to prevent those biofilms. Super smart, and definitely, where we need to be headed as an industry. But even for our small processors who maybe are not going to go toward AI and, risk-based assessments, formally, I think things like monitoring for indicator organisms really, helpful in the sense of, if I find Pseudomonas in my finished, in my pasteurized milk, it's a good indication that I have a biofilm in my system and I need to go back to sort of the drawing board, right? Taking inline samples, really helpful for sort of narrowing down where that contamination is coming from. And I think, Terry, you did that in your farm study, right? You were like taking samples at different points in the process. So there's just a lot of opportunity, but you have to kind of know that you have a problem in the first place, right?
Yeah.
And do that sort of investigation based on the fact that there is evidence that those biofilms do exist, either from swabbing or from testing the finished product or the.
One point also that De Musso and colleagues bring in their review, I think in their study is really interesting. It's that effective biofilm management require a comprehensive approach that integrates equipment design. So the
Absolutely.
company should keep in mind that biofilm likely that it's difficult to clean. So why not building a milking machine that are less cracks for biofilm formation? But I think this also will help. And also, as in Goat's review, there's an interest in using natural antimicrobial compound that can be more efficiency against biofilm. So I think this could be also interesting.
Yeah, for sure. There's also different types of stainless steel, right, that are a little
Yeah,
more
also material.
resistant, material science kind of direction.
So before we leave this question, I was reading on one of these articles about sort of stuff other than acid to deal with biofilms, and they're basically, I'm just looking at, they're talking about proteinase K,
Yeah.
maybe sort of different enzymatic treatments. Is there any hope for anything other than acid, or is it make sure the real strategy is let's make sure we get a good acid wash and call it good? Or I mean, can you add like proteinase K? That's a totally different approach, right? And so before I'll let you guys answer that question, I just want to mention we're talking about biofilms, and we can't just like put some antibiotics in the wash water, okay?
No.
Because we don't add antibiotics to milk, and if we're making cheese, we don't want to kill all the good guys, okay? So so if you're thinking, why don't they just dump a bunch of penicillin in there and call it good? Okay, well, that's not how it works, right, Nicole?
It sure isn't. That was not advocated for on this podcast.
Ever. Okay. It's a digression, all right. But not everybody is familiar with the fact that we are so careful. We never would ever allow antibiotics in the food chain. We test for it repeatedly. What we have found is that year after year after year, there are a tiny number of tankers that may come in with antibiotic residue in them. I mean, it's tiny. It's lower every year. I bet you it's under 100 nationally, annually. It's just so rare.
Yeah.
So we're not going to do antibiotics, but there are other things that are not antibiotics that maybe would dislodge these things. But are they just too expensive, just unmanageable for a sort of a large processing plant? I mean, I noticed there's a table, they mentioned some of these studies.
You want to go, Tara?
Yeah, we can kind of do it together.
Yeah.
Yeah, so one of the big things is they are new and they are expensive for different processing facilities compared to sort of the traditional acid. I think at some point we might be able to integrate more than just acid, because bacteria and biofilms, they are very smart creatures, as you will. And they do eventually kind of build up forces that make them more efficient at not being removed by sanitizers or something along those lines. So I think there is a movement towards integrating new technologies like that, but I'm not sure where we are and when it'll become the mainstay of dairy in either the United States.
Yeah, and exactly. I think the main problem is the cost. It's really expensive to produce enzyme in sufficient quantity to remove the biofilm. I think what we will see is a molecule that prevent biofilm formation, so that are not antibacterial, but just stop the biofilm formation, so the bacteria will not adhere on the surface. So in the GOTS review, they talk about quorum sensing blocking, but it's still new. So I don't think now it's applicable, but I think it could be.
Yeah, that's they're tough little buggers, and particularly when you're dealing with a food supply in the food chain, you can't apply the same approaches as you might be able to apply outside the food chain, for example.
Yeah. And I think it's why we also seeing new strategy about positive biofilm. I think, Nicole, you talked a little bit about that previously, but by preserving beneficial bacteria or by inoculating surfaces with positive bacteria that will compete with spoilage and pathogenic microorganisms. So I think this will be also another emerging solution.
So I've learned a ton today. I want to give you just a ton. I mean, a ton. I'm a dairy reproductive biologist, and now I got to go look for biofilms inside the uterus. I don't know.
You should. You
I
should.
should a biofilm for that would be so cool. Nicole, let's do a paper together. Like,
Let's do it. I'm in.
oh man, that'd be so cool. They're probably all laying on the uterine surface, those darn little biofilms. That's a fascinating idea. Those darn
I,
biofilms.
you know,
That would be so cool. That would just be so cool. And
microbes rule the world, so why not?
yeah. We always, we think about sort of the mucosal layer, the intestine, and the bacteria that sort of live in there. And but I'm going to have to get a stronger microscope because I'm always looking at mammalian cells and those little dudes. I once did some gram-negative staining on some of my histological sections from the uterus, and I was like, where the hell are the bacteria? And then I was working with a microbiologist, and they said, it's that little tiny speck. That is what you're looking. What? I thought that was dirt on my slide. No, those are bacteria, right? I don't know how you guys do it. Oh yeah. So I'll just ask you we had some final questions, and we just summarily, what do you like to do outside of science? What do you enjoy doing outside of science? Do you have any interesting hobbies or interests?
Yeah, I really love to trail running.
Oh yeah.
Yeah.
Yeah. All right. So and where is this ACER Institute? What city is it in?
Saint-Hyacinthe. It's near Montreal.
Oh yeah. Okay. Fantastic. Oh yeah. It's beautiful running up in there, isn't it?
Yeah, but before I was in Quebec City and there was a bigger mountain, but it's hill, but yeah.
Yeah,
but
yeah.
It's still a little bit new for me here, so I need to explore more.
Yeah, it's a great thing. I mean, it's fantastic. You should come to Missouri. Last week it was 98 with, I don't know, 98 degrees Fahrenheit. I don't know what that's in Celsius. I don't even know if the Celsius scale goes that high, but anyways, it's so hot. And then the humidity. So Tara, I bet you're sort of into the outdoors. Did you grow up there in Thunder Bay?
No, I grew up in southern Ontario in a really small town in between London and Chatham.
Oh wow.
Big
Okay.
agriculture town, but I like the outdoors. I spend a majority of my time golfing, honestly.
Oh. No way, really?
Yeah.
I was watching the golf tournament. Are you watching the PGA Championships this weekend? Do you follow golf on television, or you just like go golfing?
I follow it a bit. I try not to get too into it. I find myself yelling at the TV at that point. I could have made that shot, but they didn't, knowing I couldn't make that shot. But
the last round was phenomenal. I don't remember the name of the golfer, but he chipped out of the rough and sunk this putt, and it was massive distance. It was massive. It was unbelievable. Yeah, yeah, probably. So so that's fantastic. Southern Ontario near London, you must have really wanted to get out of town if you went all the way to Thunder Bay. I mean, come on, buddy. There wasn't anything closer. You gotta fly across all the Great Lakes.
Yeah, I growing up in a town of like 1,000 people, sometimes you just want to go see what else there is out there. So yeah, I picked about the farthest university I could in Ontario and hopped on a plane and just went.
Yeah, that's fantastic. Well, you guys have been great. And I always, again, as I work on the production side, and I always have a good time with the processing people. You guys always have such nice sense of humors, and I guess when I was at Cornell, I know I didn't take any food science classes. I took microbiology, but
it's not too late, Matt. You can come back.
You can come back. It's some sort of night classes for old people, but I always enjoy all that. So it's been a fantastic podcast. You guys have been wonderful guests. I'm going to encourage all my production people to listen to this podcast. It's been so fantastic. So thank you very much, Marilee, Nicole, and Tara. This has been awesome. I really appreciate your openness and how much fun you were in this. All right, everybody, that brings us to the end of our show. If you enjoyed Dairy Digressions, be sure to like, subscribe, and rate us, and spread the word to a friend or colleague, both on the food side and the production side. Right. If you have a feedback for us, please let us know at adsa@adsa.org. For show notes and more episodes of Dairy Digressions and to learn everything you want to know about ADSA, go to adsa.org. Until next time, thank you everybody for listening. Thank you.
Thanks
Thank
so
you.
much, guys.
Do you have publishing plans in the year ahead? Earn serious savings on article charges and put more funds back into your research budget with the Journal of Dairy Science and JDS Communications Loyalty Program. Reviewers and corresponding authors earn points towards discounted article publishing charges. Visit adsa.org to learn more.